Anatomy of the ear region. External ear function equals collect sound structures. Auricle or pinna. Elastic cartilage covered with skin. External auditory canal. Curved one tube of cartilage and bone leading into temporal bone. Ceruminous glands produce cerumen equals earwax. Tympanic membrane or eardrum. Epidermis, collagen and elastic fibers, simple cuboidal epith. Perforated eardrum, hole is present. At time of injury, pain, ringing, hearing loss, dizziness. Caused by explosion, scuba diving, or ear infection. Middle ear cavity. Middle ear cavity, air-filled cavity in the temporal bone. Separated from external ear by eardrum and from internal ear by oval and round window. 3 ear ossicles connected by synovial joints. Malleus attached to eardrum, incus, stapes attached to membrane of oval window. Auditory tube leads to nasopharynx. Helps to equalize pressure on both sides of eardrum. Inner ear, bony labyrinth. Vestibule. Canals. Ampulla. Bony labyrinth equals set of tube-like cavities in temporal bone. Semicircular canals, vestibule and cochlea lined with periosteum and filled with perilymph. Surrounds and protects membranous labyrinth. Inner ear, membranous labyrinth. Membranous labyrinth equals set of membranous tubes containing sensory receptors for hearing and balance and filled with endolymph. Utricle, saccule, ampulla, 3 semicircular ducts and cochlea. Cranial nerves of the ear region. Vestibulocochlear nerve equals CN8. Cochlear anatomy, 3 fluid-filled channels found within the cochlea. Scala vestibuli, scala tympani and cochlear duct. Vibration of the stapes upon the oval window sends vibrations into the fluid of the scala vestibuli, tubular structures of the cochlea. Stapes pushes on fluid of scala vestibuli at oval window. At helicotrema, vibration moves into scala tympani. Fluid vibration dissipated at round window which bulges. The central structure is vibrated, cochlear duct. Section through one turn of cochlea. Partitions that separate the channels are Y-shaped. Vestibular membrane above and basilar membrane below form the central fluid-filled chamber, cochlear duct. Fluid vibrations affect hair cells in cochlear duct. Anatomy of the organ of Corti. 16,000 hair cells have 30 to 100 stereocilia, microvilli. Microvilli make contact with tectorial membrane, gelatinous membrane that overlaps the spiral organ of Corti. Basal sides of inner hair cells synapse with first-order sensory neurons whose cell body is in spiral ganglion. Physiology of hearing. Auricle collects sound waves, eardrum vibrates. Slow vibration in response to low-pitched sounds. Rapid vibration in response to high-pitched sounds. Ossicles vibrate since malleus attached to eardrum. Stapes pushes on oval window producing fluid pressure waves in scala vestibuli and tympani. Oval window vibration 20x more vigorous than eardrum. Pressure fluctuations inside cochlear duct move the hair cells against the tectorial membrane. Microvilli are bent producing receptor potentials. Overview of physiology of hearing. Hair cell physiology. Hair cells convert mechanical deformation into electrical signals. As microvilli are bent, mechanically gated channels in the membrane let in K+ ions. This depolarization spreads and causes voltage-gated Ca+2 channels at the base of the cell to open, triggering the release of neurotransmitter onto the first-order neuron. More neurotransmitter means more nerve impulses. Cochlear implants if deafness is due to destruction of hair cells. Microphone, microprocessor and electrodes translate sounds into electric stimulation of the vestibulocochlear nerve. Artificially induced nerve signals follow normal pathways to brain. provides only a crude representation of sounds. Physiology of equilibrium, balance. Static equilibrium. Maintain the position of the body, head, relative to the force of gravity. Macular receptors within saccule utricle. Dynamic equilibrium. Maintain body position, head, during sudden movement of any type, rotation, deceleration or acceleration. Crista receptors within ampulla of semicircular ducts. Vestibular apparatus. Notice, semicircular ducts with ampulla, utricle and saccule. Otolithic organs, saccule and utricle. Thickened regions called macula within the saccule and utricle of the vestibular apparatus. Cell types in the macular region. Hair cells with stereocilia, microvilli, N1 cilia, kinocilium. Supporting cells that secrete gelatinous layer. Gelatinous otolithic membrane contains calcium carbonate crystals called otoliths that move when you tip your head. Detection of position of head. Movement of stereocilia or kinocilium results in the release of neurotransmitter onto the vestibular branches of the vestibulocochlear nerve. Crista, ampulla of semicircular ducts. Small elevation within each of 3 semicircular ducts. Anterior, posterior and horizontal ducts detect different movements. Hair cells covered with cupula of gelatinous material. When you move, fluid in canal bends cupula, stimulating hair cells that release neurotransmitter.